Hi, so my name is Darlisha D'Ambert, and for exam 3C video discussion, I'll be talking about negative regulation of B cells. So, kind of give like a brief introduction of it all. Um, we know that antibodies responses are typically triggered by the binding of antigen to B cell antigen receptors, also known as BCR. Um, the strength of resulting signals, those are determined by the outcome of the responses, and those usually vary depending on the induction of tolerance to its production of the amount of antibodies that are consumed. So going into negative regulation, we know repressor proteins, they bind to its operator, preventing a gene from being expressed. So, in other words, it refers to the control mechanisms that have matured to ensure that B cells proliferation is then slowed down. There's also an elimination within the process of negative regulation of B cells. There's an elimination of pathogens. So B cells are then entered into apoptosis, which is the death of the cells. Um, these B cells, they're responsible for humoral site of immunity. So, um, so if there's an elimination of pathogens, those B cells will then die off. CD5D1 cells, they're unresponsive to mediated cell growth systems. So when that usually happens, those CD5D1 cells will then undergo apoptosis as well. B cells from Lin kinase deficiency, they enhance proliferation response. So in other words, those B cells are mediated and are also cross-linking. B cells that are mediated signaling are in B1 cells. So they have I'm sorry about that. So they have a negative regulation by the co-receptors CD22, CD72, and CD5, which you're going to see right here in this image of cellular activation. A little about the Lin kinase. They're part of phosphorylation. So when there's an absence of the co-receptor CD22, there's a lower signaling threshold of the BCR cross-linking. So it has a major influence on the livelihood of B cells when it doesn't have its co-receptor. The subpopulation of B cells are expressed by Lin kinase 49, also known as LY49. The LY49 is a MHC class 1 regulatory receptor on the natural killer cells. There's also a FAT2 region fragment, and they are anti LY49 antibodies where they block negative signals in B cells, in B1 cells. The two co-receptors, CD5 and LY49, they also play a significant role to B cells in its regulation. So BCRs and B1 cells, they have a negative regulation by its pathways of the Lin kinase activity. So it causes, in result, it causes a negative reaction out of that. I'm so sorry about my camera. As far as research goes, there has been several experiments that have been conducted on animals such as mice for the human development for the study of controlling self-activation and differentiation. I actually seen a study, an article on NCBI, which I probably get most of my examples from in my videos. But it talked about why NWASP, it's NWASP, I say NWASP for short, is essential for negative regulation of B cell receptor signaling. So what they did was they used, I'm going to be kind of brief about it, but they used genetically manipulated mice models in total internal reflection microscopy. So they kind of demonstrated that neuronal Wiskott, I'm not sure if I'm pronouncing it right, but Wiskott-Aldrich syndrome protein, which is in WASP. What it is it's a it's co-expressed with WASP, the Wiskott-Aldrich syndrome protein in all of the immune system, and it's also very essential to B cell signaling regulators. So what it does is the gene deletion, it causes an enhanced and prolonged BCR signaling, and it's elevated levels of autoantibodies in the mouse serum. So it shows an increased signaling in the NWAS. So long story short, the results that were found was there was a new mechanism for negative regulation of BCR signaling, and it also suggests that an actin mediated mechanism for signaling down regulation. So it's kind of cool how negative regulations of B cells and how they can kind of relate to other things outside to help us find these to help us with research and also with B cells of regulation. So that was just something kind of cool that I found. But that is it, and I do not mind if you use this video.